This is our final session, as we've been doing for the last two games. All the wise people will parade from the podium, and we start first. Well, topics are not that restricted to treatment modalities or diagnostic. They are more wide, and I give the word to Karina to present the first speaker.
Okay, thank you. It is my honor to co-moderate the last session with Professor Xenos. I will start with the lady of the group, Professor Gina Brown. She has already impressed us in the morning with her lecture, and I'm sure she will present again a very surprising lecture.
Yeah, well, thank you. I'll try not to upset any more people today, but this one perhaps may provoke a little bit more upset, but it's really not intentional. So, what do we mean by omics? So, radiomics is a great term. You're probably seeing it or you're hearing about it by all the very excitable radiologists, because it makes us sound like we're scientists, which maybe we'd like to be. In fact, it borrows its name from the study of the DNA, and you've heard a lot about characterizing tumors using genomics and characterizing the proteins that these tumors make or that we make by proteomics. And so why not characterize the imaging and call it omics? Why not? And of course, we have the great power of machinery and computers to help us do this now. And what you can do, and this essentially is a summary diagram taken from radiology or Nature, and essentially every image has got a lot of information. We, as radiologists, discard most of our information. It includes the Hounsfield units, it includes the variation in the amount of skewness, kurtosis. There's a mathematical, essentially images are mathematics. They are essentially a mathematic data set, which you can crunch the numbers ad infinitum. You can produce many, many scales, many, many types of analyses, some of which are used to produce the image that we see, but others, bits of information, are lost in the computer. So the very clever people are taking all of that information and drawing various curves. And essentially what you end up with is a color map. And this kind of color map down here is just an illustration of, I don't know if this pointer works, it's just an illustration of what can be achieved by just putting a region of interest on the area of the treated tumour, for example, and then looking to see what kind of signal you're getting. And then you can data mine, and you can create a model. And you can imagine if you've got 1 million data items, you could create many models, and you can keep looking and modelling. And, for example, you can rebuild the tumour shape by drawing curves and figures around the tumour itself. You can extract items of information from the region of interest, and you can produce a radiomic features map, just like a gene expression map. And in fact, they're almost identical. So, what then happens is you have something like this, which I'm sure the scientists, the genetic scientists will know are very familiar with. You get hotspots, and you can correlate that against outcomes. And the next slide is perhaps the thing that's going to upset people. If you have an infinite number of data points, or 1 million data points, just like the chimpanzee, he will create Shakespeare's plays, and we'll create a nice marker, a nice radiomics biomarker for prognosis. And the problem, as we have found with the genetic biomarkers, is everyone around the world will be doing this with their machines, with their data sets. They'll produce their superb omics code, and then somebody else will try that, or spend a bit of money and purchase that algorithm, and try it on their data set and find that it doesn't work. And then somebody else will try something better and say, well, we've done it better, and it won't work. So that, I think, has been the problem with omics. And I think there's an added limitation for imaging. And I think as well for when we try this exercise in just looking at a single thing, which is the tumour and its imaging features, we don't take into account what the patient's doing. And I think that is often a problem. So we've been here before. I think imaging lends itself, particularly CT and MR imaging, lends itself to data analysis because there is raw data, there is numbers, there is figures that actually people who are purists about imaging will say that the radiologist is a crude surrogate for what's actually going on, which is a vast computer-derived processing approach of the images that we're capturing. And for someone like me, who sees the imaging and the pathology of tumour, this doesn't sit easy, because it feels wrong. So this is why I'm giving you this very alternative view, because this is not what you'll hear from the scientists, from those who are pursuing all of this. But my argument, I suppose, is there's a lot of money being spent on this, a lot of resource, a lot of PhDs, and you get these kind of results. I think you've all seen them in the papers if you look for them. But the worrying thing is not so much that this correlates with this, it's that the error bars, the standard, they completely overlap. So what does that mean in clinical practice? Does that mean if you have a point here that you're a good or a poor responder? What does it mean? And I think for an individual patient, we still haven't got any further forward with this kind of numerical analysis, because the data is too dirty, really. And I think, you see something like this, but then you look at the paper and they drew an ROI over this. And then you look a bit closer, there's not even any tumour on that slice. So they've derived a whole pile of numbers, data, from a badly drawn ROI. And that the same thing can happen with genomics testing. You take the wrong bit of the sample, you say that's the tumour, no one's validated it, you get dirty data. And I think this is the problem, that we rely too much on the numbers and we don't use the brain, unfortunately. So here is the sort of thing that you see in papers. These are very high-impact journals. Some of them got into Nature, some of these papers. And they all say the same thing. This would be a very promising use of imaging in the future. We'll be able to distinguish pathological complete response from none. But they derive the ROC from the same data set and then validate it in the same data set. So of course, it's highly accurate for that data set, but you try it in another data set and you don't get the same results. And again, this always worries me, these overlapping data points. It means that this is inherently a faulty technique. So, this is the problem, and every paper I've looked at suffers the same problem, which is overlap between the two, and they derive these things. So I suppose for us in the room, we need to have a checklist before we accept the consequences of radiomics, which I'm sure is going to hit us. What is the clinical need? What is the question being answered by the radiomic technique? What is the true degree of error or deficiency of the existing methods? So actually, what is the added value if you're going to put all this effort into something? Does it have a predefined threshold, and then is it validated in a new data set, in an independent data set? If you look at all the quantitative radiology research that has happened to date, not a single one has fulfilled those criteria. Not a single one. And that means that if you add up the costs of all those scans, the PhDs, it's a lot of money. And I would say that it's been a huge disappointment over these last 20 years. So, for me, common sense prevails at the moment. Lymph nodes on MRI do not predict recurrence because of the man sitting in the second row and colleagues that have followed a very important thing of taking care of the specimen. That's why this doesn't work, but this does. This is MR staged, EMVI and T stage. And then, we hear about the fact that maybe radiologists shouldn't be doing it, and maybe the machines should be doing it using radiomics, and maybe we'll get to that point. But in the meantime, I don't think the radiologists should stop trying to be good and not waiting for the machines to take us over. Because actually, at the moment, no genomics or radiomics test can beat the visualization of certain prognostic factors. And at the moment, we're using those to improve survival. So these are the sort of trials that we're doing in order to promote quality of life and good care for patients with rectal cancer. Thank you.
Thank you for your excellent lecture. You don't seem so optimistic about radiomics.
Well, I think, as Soren said earlier today, a fool with a tool is still a fool, and I think you've got to use these things responsibly. You know, just because you can get a texture from a tumour, and if you look at the variables sufficiently, you'll find some correlation. Doesn't mean to say that there's a basis for it. And so I think you have to be very sensible about how you use this kind of data.
So are nowadays ongoing studies for colorectal cancer or not?
Well, I think that in the studies, for example, we're talking about the early rectal cancers, and we're going to be training radiologists to spot an early rectal cancer, to treat them, let the surgeon deal with them, having proper information. Basically, these are the sort of things that we're going to be doing in the future. And I think that the key, actually, is rather in, you should invest in radiomics, maybe we should spend some money on it, but I think also we ought to invest in training radiologists and making sure that they're doing the best, because actually, in analysis of radiomics, if we're going to look at how we do this, we need to be sure that the input is accurate. Because if you're training a machine or any AI process to look for patterns, to look for features that may actually help us as radiologists in the future, we need to put proper verified information in order to get meaningful data out. Because at the end, machine learning is only as good as the people that teach the machines and who program those codes.
This is correct. You want to ask? Yes, please.
Sorry, Gina, it's a very simple question.
My understanding, I'm trying to get my head around some of these things, led to one discovery, and that is the criteria you use for that grey mottled-looking thing which we recognise as a cancer, and the scar at the black end on the T2 weighted image, that there is no numbering system at the moment available. Could there be?
So of course, on that level, if someone was to sit and outline the tumour on these images and feed it into the machine and say it's the tumour, then the tumour will believe what you're telling them and then start to make a measurement of those analyses. Now the problem comes when the person perhaps doesn't realise what they're looking at, puts something on and says that that's tumour, like they did in that other example, and then you get nonsense. So it's the precision with which we first delineate what we're doing. That's really the crucial thing in terms of then getting the numbers that are meaningful. I have no doubt that in 10 years, or maybe less, machines will be able to diagnose and stage rectal cancer and spot EMVI and N1C. They will, but it will be incumbent upon us to move forward and to say, well, okay, we've got this, but we need to now understand what the next step is. So as radiologists, and we're probably the first to go, then probably the pathologists will be the second, we have to work much more closely with our medical and clinical teams, our surgical teams, in order to make things meaningful for patients. Because at the end of it, it's still a tool, and we're still doctors, and we still need to understand what the basis for our decisions are. And the machine won't be able to do that, but the machine will certainly help us measure the tumour, measure the depth precisely, but we still have to pose the questions, and we still have to integrate what we know about the biology of the disease, the management of these patients, with those measurements.
Gina, I absolutely agree that it would perhaps at the moment be better to put the money into the training of radiologists or to employ many more radiologists to give the clinicians proper statements about the MRIs. But if my son were here, he would say, Alpha 1 needed only 4 hours to learn chess and to be world champion.
Yes.
So I think it might go in that direction.
I'm certain it will. I'm certain it will. But in the end, the machine is only as good as what we feed it. And we have to keep learning in order to feed it ourselves, because the machine isn't going to derive new ideas about the management structure and how the pathways and the MDT decisions and the sort of things that actually make work challenging for us. The arguments that we have today, a machine couldn't do that. So that's what I'm saying, is that there is a role for both. And probably the radiologist's life will be easier and more homogenized when we have these kind of tools, but we still have to know how to use them.
If there are no other questions, we would like to thank Gina Brown for her exceptional talk, and let's see what the future will bring.